Welcome guys to another installment of Life Sciences. Grade 11s, we're looking at photosynthesis today. Over the past couple of weeks we've looked at the basic concepts around photosynthesis, and then we looked at some investigations. But today we're looking at how to interpret some of the data and some of the results of investigations to look at how factors affect or influence the rate of photosynthesis. So let's get straight into some of the main key concepts that we're going to be dealing with in this experiment, or in this lesson, rather. So we're looking at how do we interpret data from photosynthesis investigations. So again, an overview, we've looked at photosynthesis. We know that light and water are essential parts of photosynthesis. We need light, we need water, and all that occurs in the chloroplast, and it produces starch and carbon dioxide as a waste product. And we also looked at how those factors can be influenced in the previous lesson. Today we will look at how those factors affect the rate of reactions. Some important terminology, guys. We need to look at independent variables, and as I said, independent variable is the variable that you are actually manipulating, or ask yourself, what are you testing in this experiment? And that is often what the answer should be giving you, the independent variable. The second variable that we often need to remember is the dependent variable. And the best way to remember that is, what have you measured that helps you draw conclusions? So what you measure to draw a conclusion. So what is that set of values that you, or variables that you measure that helps you draw a conclusion? And the answer is often the dependent variable. Your control variables are what have you controlled in this experiment or kept constant that allows you to get reliable, fair and consistent results. Hypothesis, guys, it's a statement of intent, and what have you predicted, or it's an educated guess. So what have you predict, or what's the prediction that you can make? So it's a statement about the prediction that you possibly anticipate happening in an experiment. Compensation point, guys, we're going to discuss this, but it refers to that point in a plant when the rate of respiration equals the rate of photosynthesis. At that net point, the amount of carbon dioxide produced by the plant during respiration is sufficient to support the process of photosynthesis. So we see at that point the net production between the amount of carbon dioxide by respiration is just enough for photosynthesis to occur. So the net production is zero. And iodine we discussed previously. We know that iodine is the reagent that is used to test for starch, and we looked at sodium bicarbonate and sodium hydroxide as being two reagents that both supply carbon dioxide and one that removes. So sodium hydroxide removes carbon dioxide from the area, and this is a source of CO2. So it's a source of CO2, and that removed CO2 in most of the experiments. So we've got to remember these key terminology or concepts that we're going to apply in our investigations. Again, we're going back to the key concepts, and this equation is something that you must be very familiar with. Learn the equation and remember what is needed for the process of photosynthesis and the products that are produced in the reaction. Cool. Remember that photosynthesis is a process in which light converts water and carbon dioxide to glucose, and glucose is a type of sugar that is stored in the plant in the form of starch. And plants use glucose for food during the process of respiration and for growth. And that plants are autotrophs, and that they're able to manufacture glucose, and that the heterotrophs are the ones that consume the autotrophs because they cannot manufacture food by photosynthesis. The key concepts that we need to discuss would be that there are three main factors that influence the rate of photosynthesis. In our previous lesson, we discussed that light, temperature, and carbon dioxide all influence the rate of the reaction, and these can be illustrated in three simple graphs where we see the trend between the light intensity increasing up to a specific point, after which any further increase in light intensity does not affect the rate of photosynthesis. And that rate of photosynthesis can be measured by looking at the amount of oxygen bubbles produced, or even determining the presence of starch and the amount of starch present in the leaf. And often, for a simplistic reason, we look at the amount of oxygen bubbles or the volume of oxygen produced as a measure of the rate of photosynthesis. The second factor that influences the rate of photosynthesis is the carbon dioxide concentration. Again, carbon dioxide concentration increases, it increases the rate of photosynthesis. After a specific point, any further increase in carbon dioxide means that the rate of photosynthesis remains optimal and will not change. And the third factor, which is temperature, we know that as temperature increases, the rate of photosynthesis increases, but it reaches an optimum temperature, after which any further increase in the temperature causes the denaturing of enzymes and hence the decrease in the rate of the reaction. And hence, because photosynthesis is controlled by enzymes, any factor that is influenced by temperature, such as enzymes, will denature and it will cause a decrease in the rate of the reaction to a point where the plant no more is able to photosynthesize and produce even starch or oxygen bubbles. So that's the gist of the basics that we need to remember and apply in the context of the various investigations and interpret that so that we can understand the investigation. So let's look at the first activity. A group of learners set up an experiment to test the effect of light on photosynthesis. This experiment was to test the effect of light on photosynthesis. They counted the number of bubbles given off from Elodia, which is an aquatic plant, as from the plant the lamp was moved away. So essentially what happened in this plant, it was set up. Here we've got a funnel that is inverted. It's placed over the Elodia plant, which is kept in water, and the inverted funnel has a little test tube over it, and these bubbles are gradually given off, and as they're given off they tend to displace the water and accumulate here. So here we've got air that is accumulating in the upper end of that test tube, and as the bubbles are given off, it starts to displace the amount of water, and eventually at some point you're going to find that the volume of air increases and the level of water decreases. So based on the volume of air collected, it could be an illustration of the degree or the rate at which photosynthesis occurred. And the variable that they've manipulated here is the light intensity, and we see that the light source was moved further away, and the intensity of photosynthesis was measured by counting the number of bubbles. Cool. So let's look at some questions. Which of the following graph best shows the expected results? So we know that we've just looked at as the distance increases, the rate of photosynthesis would decrease. If we compare that to this illustration here, it shows you as the distance increases, the rate increases. That's incorrect, and this is incorrect as well. And here it shows you a direct relationship where it increases as the distance increases, but that's not true. We know that as the distance of the light source increased from the light source, the rate of photosynthesis should decrease. So A is the correct answer there. B, provide a detailed explanation for the above answer, and as I discussed, we said that as the distance from the light source increased, the rate of photosynthesis decreased, the number of bubbles decreased, thus also decreased, and hence we see that relationship illustrating this decrease in the rate of photosynthesis. Let's move to the next activity. Here guys, we're studying the apparatus which is used to investigate the factors essential for photosynthesis. Here we've got a plant, here we've got a test tube with a rubber stopper around the neck, and it's just a plain leaf with no other factor influencing the photosynthesis. In test tube B, we've got a leaf, and in that sodium hydroxide, NaOH, and remember that NaOH removes the carbon dioxide. Here we've got black lining on the external surface. So this leaf is enclosed in a test tube which is coated with the black lining, and hence if we think about it, it means no light is going to enter that test tube. And the fourth test tube D, has silica gel, and that silica gel absorbs water. That means here we don't have no water for photosynthesis to occur. So here we've got 4 different leaves on the same plant which is subjected to different conditions. Let's look at some of the questions that possibly relate to this experiment. After a few hours in the sun, which two leaves will give positive results with the iodine test? Let's go back. So we're going to tick against the leaves that will test positive. So the question is, will photosynthesis take place in these leaves? So which two leaves will test positive for photosynthesis? And I'm going to go with this. Here we've got, it's transparent, it's nothing in it, so definitely in this, yes, photosynthesis would occur. Sodium hydroxide removes carbon dioxide and hence no photosynthesis there. No light, so definitely no photosynthesis in that test tube. And here we've got silica gel, which actually removes the water. But what you've got to remember, guys, is that water is still able to be absorbed from the xylem to the leaf, and hence definitely photosynthesis would occur in both A and D. So it will be both in A and D, as we see that light is necessary as well as it's able to get the water from the xylem vessels. Question number 2. Which test tube represents the control apparatus? It's going to go back to that. Which would be the control apparatus in this? And guys, the control for me would be C, as the black lining prevents any light from entering and hence there is no photosynthesis occurring there. And we're looking at what's the aim of this investigation? To investigate the factors that are essential for photosynthesis. So the control in this experiment would be where there is no light. Cool. Let's look at question number 3. Which of the following combinations of test tubes and aim of the investigation is correct? So we're going to look at which of the following combinations of test tubes and aim of the investigation is correct. So let's read through them and let's eliminate the ones that are obviously wrong before we attempt to find the correct answer. Test tube A, to show that oxygen is given off during photosynthesis. And if you looked at test tube A, guys, it was a plain test tube which had just a leaf and nothing around it. So it was just a simple leaf with nothing on it. Test tube B, so that's not possible because it was providing oxygen, so that's not correct. Test tube B, to show that carbon dioxide is given off during photosynthesis. If you recollect, the test tube B had sodium hydroxide in it, NaOH, and that was to show basically to remove carbon dioxide and wasn't a source. So it was definitely not that. Test tube C, to show that light is necessary during photosynthesis, and that was a test tube which had the black tin foil around, the black foil around it, and that's essentially blocking out light, and hence if we remove the light we could determine if light is needed for photosynthesis or not. Test tube D, to show that water vapor from the air is necessary for photosynthesis. We know that water vapor is not necessarily needed, but it can utilize the water from the xylem vessels, and hence we saw that the water was still coming up through the xylem vessels to the leaf, and hence test tube D, test tube and the aim is incorrect. So the correct answer for this would be test tube C, which basically shows that light is necessary for photosynthesis. Let's look at the next quest activity. Study the graph below that shows how the optimum carbon dioxide concentration changes as the light intensity changes. So here we're looking at a graph that shows an increase in light intensity, and it shows the level of carbon dioxide intensity on the y-axis. If we were to very quickly interpret the illustration here we're seeing that as the light intensity increases, the amount of carbon dioxide increases, which essentially means that the rate of photosynthesis increases. And that's something that you've got to be able to draw from this, is that the rate of photosynthesis increases as the light intensity increases, and that's purely because there's more carbon dioxide that is needed by the plant for photosynthesis. So guys, we've looked at that. I think we're going to get to some questions before I give you a short break. I think I'm going to give you some of these questions to look at, take a short break, and then we'll get back. So let's look at 2.1. What CO2 concentration will provide the highest rate of photosynthesis at light intensity of 40 units. Again, we've got to go to the graph to look at that. So if we go to the graph, we need to know at what intensity of light, sorry, I'm going to read that question again for you. At what concentration of carbon dioxide will provide the highest rate of photosynthesis at light intensity of 40 units. So essentially what we're looking at is, if we look at the light intensity of 40 units, guys, remember that you've got to use your ruler and do this. It's always better to use a ruler, and you've got to extrapolate from the x-axis and see where it intersects your graph and determine that point on the y-axis. So I'm looking at it, it's probably in intervals of 5, so between 0.8%. So the answer for that should be around that, and that process is called extrapolation. So it's roughly, sorry, I'm just going to go back. Let's go back to that graph to see if my answers are correct. So at 40 units, so I think it is, so that's fair to say that that graph is correct there, and that my possible solution here is incorrect. So I'm just going to change that, so it's roughly about 0.8%. Next question. Explain why the optimal level of CO2 increases with an increase in light intensity. And as we've discussed all the way, that the rate of respiration or photosynthesis does change when the level of carbon dioxide increases. So what is the explanation? So as light intensity increases, the rate of photosynthesis increases, and thus for more photosynthesis to occur, more CO2 is needed. And hence, guys, if we were to decide to increase only the light intensity, and you'll see that from the graph, we need to also provide the other essential elements of photosynthesis, which is the building blocks, which is carbon dioxide. And hence, as the light intensity increases, the amount of carbon dioxide utilized by that plant will definitely increase. 2.3. What would happen to the rate of photosynthesis if CO2 levels increased past the optimum at any given light intensity? So essentially what we need to determine is what would happen to the rate of photosynthesis if the CO2 levels increase past the optimal at any given light intensity. And guys, the rate of photosynthesis is quite simple. The rate of photosynthesis will not change and will remain the same, because remember that plants have a fixed number of chloroplasts in them, and hence no matter how much of carbon dioxide you provide at light intensity, they are restrained by the number of chloroplasts present in the leaf. So essentially what you're saying is that no matter how much petrol you give to a car, it cannot go faster than the amount of petrol that it can combust to produce more energy to travel faster. So essentially the analogy is that no matter how much more carbon dioxide and light you provide, it gets to a point where, from that graphical illustration, it gets to a point where the rate of photosynthesis remains constant. And that is essentially because the number of chloroplasts present in them is limited, and it will stop the rate of photosynthesis. And essentially that is the next question which we needed to explain in terms of the answer for that question. So explaining your answer would be, the rate of photosynthesis depends directly on light intensity. The light intensity determines the rate at which photosynthesis occurs. So no matter how much of carbon dioxide is supplied, the leaves can only absorb a specific amount of carbon dioxide, and hence that rate of photosynthesis would be a maximum for up to a specific point. Okay, so guys, we've looked at that investigation. It's been a long session. I want you guys to have a quick stretch break before we get back to looking at some more questions and investigations. Welcome back, guys. Hope you had a good stretch break. Let's look at another investigation. An experiment was conducted to determine the effect of different wavelengths of light on the rate of photosynthesis. That's very important to remember. It's the effect of different wavelengths of light on the rate of photosynthesis. This was done by changing the color filter and measuring the rate of photosynthesis. The setup of the experiment is shown below. So let's look at this experiment. Here we've got, very similar to the other experiments, here we've got an aquatic plant called Gr11. It's placed over an inverted funnel over which a test tube is placed, in which gas is given off, and that gas is collected there. Here we've got a stand, we've got a light source through which light enters, and that light passes through a transparent cellophane filter, which is of different colors, and these different colors essentially will represent different wavelengths of light. And we've got blue, green, yellow, and red, and we've got a colorless one as a control. So essentially what we're talking about, guys, if we go back to a basic physics, we know that monochromatic light, or the light that we see, is made up of different colors of light. And if you look at the rainbow, you would see that you've got the seven different colors, and essentially we've got white monochromatic light made up of different wavelengths of light. We cannot see that with the naked eye, but the plant, in the chloroplast, we've got chlorophyll, which are of different types, and that chlorophyll of the different types of chlorophyll absorb different wavelengths of light. So essentially what we're saying is that if we can manipulate the type of light that we subject the plants to, the rate of photosynthesis would be affected. So quite an interesting experiment. Let's look at what they've done, and let's try and understand how light can influence the rate of photosynthesis when we manipulate the wavelength or the different intensities or colors of light. Okay, so the graph below shows the data that was obtained from the experiment in which different filters were used. Now I'm going to spend a little time trying to unpack the information in this. Here we've got, on the x-axis, we've got wavelengths of light, and in different nanometers, and that's the unit which measures the wavelength of light. And here we've got, at the bottom of the spectrum, we've got violet, and that's at 400 nanometers, and we see that as the wavelength of light increases, we've got the different color intensity. So violet being the lowest wavelength of light, and we've got red having the highest wavelength of light, which is around 650 to 680 nanometers. Okay, so here we're seeing, on the x-axis, we're seeing the wavelength of light, and this graph represents both the absorbance of chlorophyll A, so that's the pigment that we're talking about. There's chlorophyll B, there's xanthophylls, carotenes, which absorb different wavelengths of light, but here we're focusing on just chlorophyll A, and that is being measured against the rate of photosynthesis. So here we're seeing the absorption illustrated by a solid line, and I'm going to try and highlight that in blue. Excuse me, and the rate of photosynthesis, and I'm going to try and show that with maybe yellow. Let's see. So that is the rate of photosynthesis, and that's quite interesting to see. Here we're seeing two different factors that are being measured simultaneously when the wavelength of light is being changed. And essentially what we're saying is that if you look at this graph, guys, if we looked at this graph here, this would be a point at which the rate of photosynthesis is optimum there, and here it would be the least. And if we compare the absorption of chlorophyll, the absorption of chlorophyll is the highest at this specific point here and there. And here you're seeing a close relationship between these two factors, between the absorption of chlorophyll A and the rate of photosynthesis at this specific wave frequency or wavelength of light, and here. So we're seeing that at two specific ranges, we're finding that the rate of photosynthesis is relatively higher compared to at this point where it is the lowest. So we've tried to understand the graph and the relationship of absorption of chlorophyll A and the rate of photosynthesis at different wavelengths of light. So quite an interesting experiment to unpack. So let's read through some of the questions and see if we can answer them. So again, what was the aim of the experiment? And guys, we looked at the graph, we looked at the intention of the experiment, and it was basically to determine the effect that different wavelengths of light have on the rate of photosynthesis. And that essentially comes across clearly in that graph. We've manipulated, we've changed the frequency of the wavelength of light, and we've looked at the rate of photosynthesis and we've measured that. So it is essentially looking at the dependent variable, which is the rate of photosynthesis, and the independent variable would be the different wavelengths of light. Good. Let's look at the next question. Why was different color filters used? And essentially, as I said, these color filters will allow white light to pass through and to filter out the rest of the light and only allow a specific color of light to pass through. And hence, when you used a red filter, it was red light that passed through. When you used a green filter, it was green light that passed through, and so forth. So these filters were used to allow only a specific color of light, and in other words, a specific wavelength of light to pass through it. So this was manipulated to allow the wavelengths of light to be manipulated or to be controlled from an independent variable perspective. So let's look at it. So why was different colors of filters used? And that essentially was used, guys, so that when light passes through different colors, the colors of light will allow only a specific wavelength of light to pass through. So it was the color of the filters was manipulated or changed so that we could change the wavelengths of light passing through the filter paper or through to the plant. The next question: at which light intensity is the rate of photosynthesis the highest? And we've got to go back to the graph, guys. So we've got to look at which light intensity is the rate of photosynthesis the highest. And if we look at the axes here we've got, I'm just going to erase some of this here. If we look at the rate of photosynthesis, where is it the optimal? And if we go back to the yellow line, we've got to go there. So it's going to be here. So it's around between 400, so that's around 400, 450 there. So it should be between 420 nanometers. So let's go straight to the answer and see how accurate that extrapolation is. So it's between 420 nanometers where the rate of photosynthesis is the highest. 3.4. At which color range is the absorption of light the greatest? So here we're looking at which color range the absorption of light is the greatest. And guys, if you go back to the graph, it's between violet and blue. I'm just going to skip back quickly to the graph. If we look at the color range at which the absorption is the greatest, it is between this. So here we see on this graph, if I were to very quickly illustrate this area and highlight it, it is in this area that the absorption of light is the greatest, and it is between both violet and blue that the absorption of light is the greatest by chlorophyll A. Cool. Identify the colors at which the rate of absorption was the lowest, and that's quite interesting. Here we've got to look at the lowest. We went back to the graph and we looked at that. Here it was the highest, and the lowest, guys, at which range of light intensity was it the lowest? So as I discussed earlier on, we're looking at this area here where the rate of absorption and the rate of photosynthesis is the lowest, and it would be around this area here. Okay, I know that my graph's looking very busy now, but I encourage you to do that so that you can look at trends, you can identify and make your answers more reliable when you do that. Okay, so here we're looking at it's between green and yellow, and hence you'll find that when farmers are actually cultivating plants in greenhouses, they're least likely to use green and yellow color intensities of light, and because essentially chlorophyll A is not going to absorb it. However, remember that there's chlorophyll B, xanthophyll, carotenes. These are also able to absorb different wavelengths of light, not necessarily green and yellow, but other wavelengths of light. 3.6. Provide a reason for your answer in question 3.5. So let's look at the reason. So chlorophyll A and B are green and yellow. I'm just going to get to my highlighters, green and yellow in color, and hence do not absorb green and yellow light. Thus, the rate of photosynthesis is the lowest at these ranges of color intensities. Quite interesting to note. 3.7. Identify the following variables in the experiment. We need to look at the independent variable, which we discussed, and the question that you need to ask yourself, what are we testing in this experiment? And the simple answer to that would be, we are testing the different wavelengths of light. So it will be the different wavelengths of light, or in other words, the different colors of light, which essentially is an indication of the wavelengths of light. The next question. Identify the dependent variable. Remember, guys, ask yourself this question. What are we using in this experiment that helps us draw a conclusion? And essentially what we've measured the absorption of chlorophyll A, and we've measured the rate of photosynthesis on the y-axis. So essentially what we're saying here, it would be the rate of photosynthesis that is dependent on the absorption of light, and hence the rate of photosynthesis being influenced. So guys, we wrapped up that question. We're going to look at another graph and another investigation. And before we get to that, I think it's very important when you look at an activity where you're testing different variables, it's important for you to understand the aim before you actually start solving the problem. Because if you understand clearly what is being tested, it makes the context of the experiment much more accessible in terms of answering those questions. So let's read the activity and try and establish what is the aim of this investigation. So an investigation was conducted to determine the relationship between light and the release of carbon dioxide. That's very important. So I've very quickly picked up that the investigation is testing the effect of, or the relationship between light intensity and carbon dioxide. Let's read further. By the leaves of plants grown in a sunny and shady area. So here we're looking at the relationship between light and the carbon dioxide released in plants that are grown in both a sunny area as well as a shady area. And we know that plants tend to grow in sun, or they love sun, and there's certain plants that tend to grow more profuse or more better, or we're better adapted to growing in shadier, cooler areas in terms of their habitat. So let's look at if the rate of photosynthesis in these plants are affected by the light intensity. So the results are indicated in the graph below. Let's study the graph and answer the questions that follow. So let's look at this graph. Let's read the heading of the graph. That's very important. Here we're looking at the change of the net carbon dioxide uptake with light intensity. So this graph illustrates how the carbon dioxide intensity intake changes when the light intensity varies. We also notice that there are two graphs illustrated. Let's look at the key. So the graph that is illustrated in orange shows a plant that is grown in a shady area. So this is a shade-loving plant, and the graph that is illustrated in blue shows a plant that loves to grow in sun. So it's a plant that is well adapted to living or growing in a sunny, bright, well-lit environment. On the x-axis, the independent variable is the light intensity. So here we're seeing that the light intensity increases, and on the y-axis we're seeing the net carbon dioxide intake. So we're looking at how much of carbon dioxide the plants absorb during the process of photosynthesis. So let's look at some of the questions and further. There's another concept that we need to look at in this, is that what intensities of light drive photosynthesis? Essentially what this graph shows you is that it shows you the relationship between respiration. Here we're seeing respiration and we're seeing photosynthesis. And at some point, guys, at this point here, which we call the compensation point, and as I said earlier on, it is at this point where the net intake of carbon dioxide by the plant equals the net amount of carbon dioxide produced during respiration. So essentially what I mean by this is that during the process of respiration, the mitochondria produce carbon dioxide. Remember that plants require carbon dioxide for photosynthesis, and that could be obtained from purely by the mitochondria producing carbon dioxide. So that relationship between the chloroplast and the mitochondria, where the net CO2 production by the mitochondria equals the net CO2 intake by the chloroplast, that specific point is referred to as the compensation point. So we're saying that here there's a graph that illustrates the compensation point, and it is essentially the point where the plant, it breaks even, where the plant produces carbon dioxide enough to be able to support photosynthesis. So I think that's very important when we look at the context of this experiment, going back to that graph. So here we're seeing, on the x-axis, we're seeing a scale that shows 0, but we're also seeing a negative scale showing the net carbon dioxide intake. That's going to be quite an interesting point of discussion when we get back after a short break. So stay tuned, let's stretch a bit and get back to the next part where we look at compensation point and how that is in effect very important to a plant. Welcome back, guys. So we're in the last session. Let's look at this experiment. Let's answer some questions on this. Quite an interesting experiment where we're looking at both the relationship between photosynthesis and compensation point in terms of the amount of carbon dioxide produced during respiration. So we did look at this graph before we went into the break. We said that this graph illustrates both two plants growing in different intensities of light. One, a plant that loves to be in the sun, and one, a plant that is growing naturally in a shady area. And we're looking at how much of carbon dioxide intake that is. And here we've got, quite interesting to note, is that that is the 0 scale on the x-axis, and we're seeing that there's a -5 and a +5 on this. So it's going to be quite an important point of discussion that you need to take note of when we answer the questions. So let's look at 4.1. At what range of light intensity is carbon dioxide released in plants that are grown in sunny and shady areas? So at what range of light intensity is carbon dioxide released in plants? So we're looking at the range. Okay, so guys, I'm going to go through the answers, so I'm going to try and explain that for you. So in a sunny plant, it is at 5 units, and in a shady plant, it is at 8 units. Let's go back to this. Okay, so at what range is it? So in a sunny plant, it starts, guys. So let's see. So here, where is carbon dioxide starting in a sunny plant? So here, if you look at this point here in a shady plant, guys, so that's around 5 units there. But if we compare that to the plant that grows in the sun, it's around 8 units there. And that is how. So it begins to start releasing or taking in carbon dioxide at this specific point here for both. And you'll notice that in a shady plant, it's significant, it's slightly lower than that in a sunny plant. Cool. We're going to constantly go back to questions and that graph. 4.2. Name the process taking place in the leaves that uses carbon dioxide. So again, uses carbon dioxide, guys, that's simple. We are dealing with photosynthesis. No marks for getting that correct. It's an obvious question, so I hope you guys would have got this correct. So we're dealing with photosynthesis. The process that uses carbon dioxide is photosynthesis. So that's a marks for jam. 4.3. Why is carbon dioxide released when the light intensity is 0? So let's go back to that. If you go back to the graph, guys, if you look at the graph here, we're seeing the light intensity is 0 here, yet we do have carbon dioxide being released. So here it's intake. Here it would be, there's still light intensity at 0, there's intake of carbon dioxide. So why would that be necessarily happening? So let's look at the answer for that. So when we look at this, guys, there's no photosynthesis occurring, and thus the only process that releases carbon dioxide is respiration. So it's because of respiration that carbon dioxide is being released. Remember that plants respire. They produce, from the mitochondria, you have CO2. And that CO2 is being released during respiration. Next question, guys, 4.4. How much of carbon dioxide is taken up by the leaves grown in the sun when the intensity of the light is 60 units? So we've got to go back to the graph and we're looking at plants taken up by Gr11 in this, sorry, let me say this again. How much of carbon dioxide is taken up by the leaves grown in the sun when the light intensity is 60 units? So I'm going to quickly click on my answers, and I'm going to go back to 60 units and try and explain to you exactly how we extrapolated. Okay, so at 60 units, guys, let's look at this. If we go back to that, sorry, I'm just going to click on this. Let me get to my highlighter, and if we go up here, guys, if we look at extrapolation, remember you've got to use a ruler. At 60 units of light intensity, if we extrapolate further up, we will find that it touches the graph around that area there, and if we extrapolate onto the y-axis, we find that it uses approximately 20 units of net carbon dioxide. So the answer there was 20, and that was purely for extrapolation. What you've got to try and do is, guys, you've got to show your extrapolation, show that on the graph paper. Showing off your extrapolation often means that you can get a mark if it was a 2 mark. So show off the extrapolation, show that using your ruler and exactly how you arrived at this answer. Often your teachers are looking for the process or the methodology as to how you've got the answer. We're going back to some more questions relating to this. When the light intensity is approximately 10 units, so when it's 10 units, there is no net change in the concentration of carbon dioxide surrounding the plant grown in the sunny area. give an explanation for this. So we've got to explain why at 10 units there is no net change. I'm just going to go back to that guy. So if you look at 10 units, there is no net change in the amount of carbon dioxide being produced. And essentially I said that it's got to do with the compensation point, which is essentially the point at which the rate of photosynthesis and the rate of respiration are equal. So when we look at this question here, guys, it talks about essentially the amount of carbon dioxide given off is equal to the amount of carbon dioxide taken in, and we refer to that point as being the compensation point. I'm just going to take you back to this graph here. So this is what I'm referring to. So at this specific point, it is where the amount of carbon dioxide produced by the plant during respiration is sufficient to sustain the amount of oxygen, carbon dioxide needed for photosynthesis. So that is the point which we refer to as the compensation point. Quite a nice question. Please remember that. It often comes in, it's your higher order thinking questions that need you, that tease you and often need you to be able to answer them with greater depth in your answers. Okay, we're going to spend some time on this next question. At what light intensities above 25 units in plants grown in a shady area, the amount of carbon dioxide remains the same? Suggest an explanation for this observation. So we're going to look at, I'm going to read this for you. I'm going to give you 2 minutes to work through this. I'm going to switch to my graph so that you can remember this question and that you can answer them. So I'm going to go back to this question again and then to the graph. At what light intensity above 25 units in plants grown in shady areas, the amount of carbon dioxide remains the same? Suggest an explanation for this. So we're going to go back to the graph and I'm going to give you a couple seconds or maybe a minute to work through this question. So at what light intensity above 20 units does the plant continue to photosynthesize? So guys, we're going to have a 1 minute break. In that I'm going to flash back to the question and the graph so that you can answer this question correctly. I know that was a difficult question, guys. I'm going to read that question again for you, go back to the graph and see if we can answer this together. So let's look at that question again. Quite a big question to read and to remember all of that, but let's go and do it together. So the question is, at light intensities above 25 units in plants grown in shady areas, the amount of carbon dioxide remains the same. Suggest an explanation for this. So if we go back to the graph, so it says that plants grown in shady areas above 25 units, if you look at this area of the graph, there is no change in the net carbon dioxide intake. Suggest an explanation for why this remains constant. And guys, if you do look at this, it ties in with all the graphs that we've been doing since this morning. So we looked at a graph this morning. We said that as the light intensity increases, the rate of photosynthesis remains the same. Here we've got to explain why that happens. And essentially what you've got to remember is that plants are limited by several factors. One factor is light intensity, the other is temperature, the amount of water, the amount of carbon dioxide. However, you've got to remember that the number of chloroplasts in a leaf is also a factor that is going to influence the net rate of photosynthesis. So if we have saturated the plant with carbon dioxide and it cannot take in more carbon dioxide purely because it does not have any more chloroplast, the rate of photosynthesis is going to remain an optimal at a maximum potential rate. So essentially what we're saying here is that the plant cannot photosynthesize anymore, and that's essentially because one is that it's saturated with carbon dioxide. The chloroplasts have absorbed a maximum amount of carbon dioxide that they can take in, and hence any further increase in the amount of carbon dioxide will not influence the rate of photosynthesis in a positive manner. So let's look at the answer, which I've written down for you guys. So what we're saying in this is that, if you go through this, is that plants have reached a maximum rate. They've reached a maximum rate of photosynthesis. It has a specific concentration of chloroplast in the leaves and thus can only absorb up to a certain amount of light. If there is any further increase beyond this concentration, it will not influence the rate of photosynthesis and thus it remains constant. So as I said, it can absorb a maximum amount of carbon dioxide, after which the chloroplasts are saturated with carbon dioxide and will not influence the rate. So also plants that are grown in shade have fewer chloroplasts than plants that are grown in a sunny area. So if you look at a plant that is generally growing in a shady area, it is not as dark green intensity as plants that are sun loving. So you tend to find that the plants that are growing in shady areas have a lighter green color, are not as dark green in intensity, and that's purely because they do not have larger concentrations of chloroplasts as plants that are growing that are sun loving plants. And why is this important? It's very important to understand the concept of this in the context of when you look at plants and how they adapted from an agricultural perspective. So if a farmer is growing these plants and he decides that he needs to give it a whole lot of carbon dioxide, and which is quite an expensive process, he needs to understand at what specific point is the carbon dioxide that he provides going to saturate the chloroplast, after which anything further is going to be economically not viable for him. So it's important to understand that photosynthesis can occur, but at some point it will not continue to increase because the chloroplasts have reached their maximum potential at which they can absorb carbon dioxide. Quite an interesting question to look at. Let's look at the last question and we'll do an overview of this. Name 2 external factors that need to be kept constant during this investigation. So when we look at external factors, there are several factors that tend to influence the rate of a reaction. We discussed this, but let's look at the solution to this. So what are the factors that make this experiment fair? In terms of, we know that water will influence the rate of any reaction based on knowing that water is required for photosynthesis. We also know that temperature can affect the rate of the reaction, and hence in order for us to ensure that the reaction or this experiment is fair and reliable, we've got to control the amount of water that is provided. We've got to maintain a constant temperature because both water, temperature and light intensity affect the rate of photosynthesis. So guys, we've come to the end of this lesson, but I know that we've got to touch base with some of the core essential factors that we've discussed in this lesson, and I'm going to go back to looking at some of the graphs that we discussed at the start of this lesson, and those graphs were graphs that we looked at in terms of how they influence the rate of photosynthesis. So to wrap things up, I'm going to very quickly go through these. I'm going to erase this out for us quickly so that we can touch base with exactly what we've discussed in this episode. So guys, we've looked at the 3 factors that we discussed. We discussed light intensity, we've discussed the concentration of carbon dioxide and temperature. Knowing that all 3 factors influence the rate of photosynthesis, remember that light have different wavelengths, and those wavelengths of light are absorbed at different intensities based on the type of chloroplast present in the leaf. Carbon dioxide also is absorbed by the leaf, and that's the chloroplast that absorb them. Any further increase in carbon dioxide concentration can saturate the leaf and will not influence it. Hence, we reach that optimal range. And finally, we looked at temperature. We said that because photosynthesis is driven by enzymes, the rate of photosynthesis is controlled by enzyme activity. And hence, if we do change the temperature constantly, at some point it will denature the enzymes and it will cause a decrease in the rate of enzyme activity. So guys, remember these 3 factors and how we control them when we are carrying out investigations or interpreting photosynthesis investigations. From my side, I hope you've enjoyed the lesson. Take care, study smart.